अरे अल्लाह, ये सेटिंग कर लेके बैठा मैं। Guys, can you see the screen now?
Yes, sir, you are visible. Welcome
I'm,
back
I'm,
once.
Thank you. I'm really sorry for this technical glitch. And I'm sorry, Samriddhi, and for the inconvenience caused to my
No, no, no problem, sir.
students.
No,
And
no
I
problem,
heard
sir.
the introduction. Thank you, Samriddhi, for the wonderful introduction. So, before not much delaying, I just want to get into the topic. And today, topic is like drug studies, animal studies in drug development. And I will be also discussing about pharmacokinetic and dynamic parameters which are involved during the drug development phase. So before getting into the deeper, I would like to just elaborate on what is drug development. So it's nothing but a multi-stage journey, what you call it as, and right from your initial concept till your drug get marketed, I mean available in the market. So we have different stages during the drug development phase, and out of that, animal studies are one of the crucial step in your drug development phase. So are animal studies, are animal experiments really necessary during drug development phase? Um, yes, of course. Unfortunately, yes. We required animal testing, which is rigorously testing on your animals to know your safety and efficacy during your drug development phase. So, why the animal studies are required in drug development phase or to establish a drug to get a drug into the market? So the main reasons are here, like we have some international guidelines, regulations. It says that animal testing are required prior to testing the same drug in human beings. The basic concept of animal testing is properly assessed before we do a drug trial in human beings. So prior to each project, we always check whether the approved method is available that doesn't really rely on animal studies. If possible, we apply it. So we will try to minimize as much as possible where we can have the existing data. In that case, in that scenario, we will skip some of the steps during animal studies. So the good news is, like the as we are moving towards the era where we have a lot of digital and new developments in the society. So definitely in this scenario we are reducing our animal studies, and as you can see from 1980s to 2025, we have seen a lot of drastic changes in animal studies, which have reduced the number of animals used in animal studies. And by that way, we can safeguard the animals here. And definitely we have a lot of alternate options. The scientists are working on it, and we can see in the coming years that and regulatory authorities are also consistently behind where we can reduce the number of animals used in animal studies. So we have some alternate options. We will be discussing each. So basically, we have three alternate options here, like replacement. So here it is nothing but we use some computer models, simulations, and cell technology, which is under the process, and most of the companies are replacing it slowly from animal model to cell technology and computer models, and reducing reduction. Yes, definitely we were I mean, we are reducing the number of animals. You can see in the second, our subtitle here is like we are reducing the number of animals to, compensate the animal things here. So, if we improve our statistical analysis methods, in that way, we can reduce the number of animals used in the animal studies. And refinement, yes, definitely. We will also have to look into the pain and distress what we are giving during the studies to the animals, that improving their welfare can also, contribute the, reducing the number of animals in animal studies. So here one question is like everyone asks whether the animal studies comes under clinical phase or preclinical phase. Here I want to clarify you guys that this is not a clinical phase. Clinical phase start from phase 1 and till phase 4. That is your post-marketing surveillance, and the animal studies comes under preclinical phase. So as I said, like during drug development phase, we have 5 critical steps here. As I said, this is a multi-stage journey, as I said. So here, the very first step is your drug discovery and development. We will not go much into deeper on that part as we are into animal studies topic today. So preclinical is nothing but your animal studies. The other one is drug development phase. That is, you are nothing but phase 1 to phase 4, and then I mean phase 3, and then your FDA review and regulatory approvals will be in place, and then finally step 5 is your phase 4. That is your post-marketing safety monitoring phase. So before I go into deeper, we have 4 important definitions which I would like to remember by all of you guys, so that the coming slides will help you to understand. So here, what is in vivo is nothing but within the living organism. When we are doing experiment within the living organisms or within the animals, like or human beings, is called as a in vivo. Example is mice, rat, dog, animal models, or even humans are called as in vivo testing or in vivo. And in vitro is nothing but by using some equipments like petri dish, glass, your test tubes, where with a controlled environment. If you are carrying out such studies in such an environment, then the term will come as a in vitro. In ex vivo, here definitely, so we will be, as the name suggests, ex vivo. So we will explant, extract some of the organs or the tissues from the animals, and we will test our drugs on these tissues or um, organs on to know the safety, efficacy, and other parameters. And in silico, definitely nowadays, as I said, that the whole industry is now moving towards the um, a computerized model as we improve the technology. Most of the companies are now moving to in silico assays, where we use some biological experiments by using your computer or computer simulation kind of thing, where you will get the data. So, so animal studies in drug development phase is nothing but, as I said, animal experimental or animal research or in vivo testing. So why we required animal studies? Basically, we have 3 important parameters here when we are starting our animal studies concept. One is to know the safety and the pharmacokinetic parameters, and we also have to assess the proof of concept of efficacy of your new product which you are under the drug development phase. So here we have a lot of animals and used during your animal studies, and for different indications, different animals have been used. And for example, like for if you are carrying out a drug for your diabetic studies or cancer studies, we use generally rat, and Similarly, for neurological studies, you'll be using cat and vice versa. Based on your demand, we have a lot of animals and different indications. Different animals have been used, but the list is like ras, mice, rabbit, dog, cat are being used in animal studies. So roughly, you can see that half of the animals are used in animal studies are mice, followed by your rats, and then amphibian fish, rabbits, and so on. So the list goes on. So before you start animal studies, as you must have already covered in your previous topics, you required an ethical approval to do human being studies. In similar way, we required some ethical approvals, ethical regulations, guidelines for conducting animal studies as well. So here we follow OECD guidelines for animal testing or preclinical testing, or and also we required a GLP certified laboratories where you have a good laboratory practices to carry on your preclinical laboratory studies. And as I said, like here also we required committee approval, ethics committee approval to conduct your animal studies as well. So basically, two things, two main important regulatory requirements to conduct animal studies. One is your ethical approval, regulatory approval, and the other one is you follow your GLP, good laboratory practices at your lab. So, as whenever we start a step or we start a project, we definitely come up with a aim and objective. And here, the aim of animal studies and objectives are to decide the drug or device is reasonable, safe, and effective. And the objective is definitely with the data what we get into the from your animal studies, the data should be submitted to regulatory authorities to move on to other phases of our clinical trial. So aim is to know you're safe and effective, your drug or device, and the objectives are ultimately you will be submitting the data to your regulatory authorities to get the approval for further phases. So as we get into elaborate about your safety and efficacy, I can broadly classify it into mainly two types of studies here, like toxicological studies and pharmacological studies. So during toxicological studies, like we generally see how toxic the drug could be. We have a lot of different methods here to test the toxicity of the drugs, and coming to your pharmacological studies, we have a lot of parameters to be checked during this process, like the how the drug, get absorbed, eliminate, metabolize, excrete, and how the drug show its action on the particular tissue or organ. So all this will be studied during your animal studies before we get into your actual human studies. So safety efficacy can be determined by your toxicological studies and your pharmacological studies. So moving on, toxicological studies, like drug toxicity is nothing but when a drug can damage your organ or a tissue completely or partially. So that is the level where the drug concentration of the thing. So that is called your toxicological effect or toxic effect on your organism. And generally, we do toxicological studies to determine the dose response, estimation of your pharmacological and toxicological effects. So, moving on to the toxicological studies, we also assess the drug distribution to organ system. So, different drugs have a different efficacy, you know, uh, reach to reach the organs with different receptors by different models are there. Drug binding and all that stuff is there, to reach a drug from uh,
from place where you get administered till you get into the tissues. So, and also to identify the metabolic, kinetic, and elimination pathways. So here broadly, toxicological studies are basically divided into two based on the studies of what we do in animal testing, like acute and chronic. So acute is nothing but when you take a drug and you immediately show a side effect or toxic effect, like a reaction to the drug or kind of things. Or there are some drugs like, you take the drug in a minimal concentration and you can see the toxic effect, maybe a couple of days or months or years. So we have a lot of examples here. So, for example, for chronic toxicity, like if you take paracetamol for a long time, your liver get damage here. So it comes under toxic, chronic toxicity. So, and whereas your acute toxicity, you show the very immediate reaction to your drug, like hypersensitive reaction to your skin, rashes, getting blisters on you immediately after that. So based on that, we have divided your toxicology studies into acute, chronic, or short-term or long-term. So, what is acute? Exactly we do to know the toxicity during the acute phase? So, here what we do is like we will be giving a single dose, right, starting from a minimal dose like 5 mg. Slowly we escalate to 10, 15, and we see what could be the concentration of the drug can cause your toxicity to your, to your liver or to organ or to whatever it is. So we will start with the smallest amount of drug. As you can see in the slide, like here I started with 10, 15, 20, 30, and we go on. And wherein you can see the yellow is your indication that you're about to reach the toxic level, and the red one is your toxic level. You have reached the toxic level where you are damaging your organs here. So yes, this is the mg, what you can say, this is the dose which can show you the acute toxicity. So, and we go on moving on till we see the life threatening. There is nothing but a permanent change to your organ, or liver damage, or organ damage, or even death. So we till we increase our dose till we get the life-threatening conditions, and with this we will be assessing, we will be getting a good data for your acute toxicity levels. Moving on, so how much duration you will be exposed with your drug to the animals to get the toxicity data? So here generally, as per the guidelines, as per the rules and regulations, we observe until 14 days after we give the drug to the animal, and we right from the administration of the drug to the animal, we will record till the day 14, how the acts on animal, what are the toxicity, what are the side effects which are seen by the particular drug. It could be liver toxicity, it could damage your liver, renal system, it could damage your brain neurological system, or whatsoever. So we keep tracking until 14 days get completed, and or till it reaches to the life-threatening conditions. So these studies, also provide a general data and a potential drug, such as dose response relationship. So with the dose, what is the response of your particular organ or your particular thing? So we can establish relationship between dose and response to acute toxicity. So moving on to within the toxicity, we have the other step here. Like I said, a single dose toxicity. Now we are the process is same, but we do repeat doses are given in a serious manner. Like from if we give 5 mg to some group, we will be giving some 10 mg or directly to 20 mg to the other group, and we assess the effect or toxic effects of the drug on on animals. Sorry. So moving on, like for long-term toxicity or chronic toxicities, generally we see the complete damages to the organs, and we can also see there are long-term effect can also change your genes level. So you understand how the importance of your toxic studies are should be, because it can change your DNA levels as well. So, for example, here, the titanium, what you use during your dental implants, it has shown that over a period of years, 10 to 15 years later on, after using titanium, your DNA get altered there, and it is documented. So this, I mean, it's a known area here. You see, this, you are changing your uh, genetic parameters with the drug. So we have to consistently watch throughout the, with different levels, with the different time duration, how the drug responds and their toxic levels. So there are some other studies also we have to assess before giving to before moving to human studies, like carcinogenic studies, like for example, chloramphenicol is an antibiotic we generally used thing, and it is definitely associated with the leukemia, which is one type of cancer. So chloramphenicol are well known to cause cancer if it is given in higher doses or it is not monitored properly. So the other example is cyclophosphamide. So this particular drug increases the risk of again leukemia, and we have seen a lot of cases where it causes urinary bladder cancer as well. So because we are talking about a lot of toxicity, so here there are three definitions, particularly uh, we will be using repeatedly during toxicity studies, like ED, LD, and TD. So ED is nothing but your effective dose. It's nothing but your drug is in a good therapeutic range, which shows a good therapeutic outcome. So at the level at which your drug is there, it will show a good therapeutic response. And whereas your TD, definitely it's a higher dose, what beyond your therapeutic dose, and which can cause liver, organ damage, or even death can also occur with your toxic dose. And lethal dose is completely, uh, straight away, you give 1000 mg, immediately you can see the animal is no more. It can cause directly death. So it's that dose is generally termed as a lethal dose. And we have some other definition like ED50, TD50, and LD50, where ED50 is generally termed as where your total population, out of total population, 50% of your population shows therapeutic good response, and whereas your TD, your toxicity of 50% of your population shows toxic effects of your particular drug, and LD50 is nothing but a lethal dose, where 50% of your population will show the cause of your death. So moving on to further on toxicities, a generalized a table here made for your better understanding here. Like we have different formulations, right? Like we have IV, we have oral, we have parental, we have a transdermal. We have a lot of formulations which comes. The drug comes in different formulations, as you can see in your pharmacies. So different for different routes of your drugs administration have different levels of testing here. For example, if your particular drug is in oral form or parenteral form or transdermal form, the guideline says that mm, at least you should be using two species at least for 2 weeks to test the toxicity levels. And similarly, if you are using, some high-end kind of antibiotics or cancerous drugs, and then definitely the duration of your drug, the data should be more. and you will be extending your toxicity observation till 24 weeks as well. For inhalation, for a general anesthesia kind of formulation, for aerosols, we get a lot of sprays, we get a lot of inhalers. For that, you should be using, I mean, two species up to 4 weeks, or I mean, based on your indication, based on your drug concentration. So we have different guidelines here to use different animals and different time duration to know the toxicity. And for local toxicity, generally for dermal, ocular, nasal, or rectal, if your formulation is, then you are supposed to do at least for 2 to 4 weeks duration, and by using one or two species animals to know the toxic levels. So it's a broad concept here, but with particular to your subject, I have pulled out some important what I can say points here, so that you start your career in clinical research. So definitely we have a lot of other concepts as well if we get deeper into that, but when it comes to clinical research, I believe that this content is enough for you to crack your um, interview. Coming to pharmacokinetic and pharmacodynamic topic here, guys, as I said, for each drug, we should know the properties, how it absorbs, how it metabolizes, how it distributes, and how it gets eliminated from the body. And the dynamic parameters are definitely how the drug shows the mechanism of action of your drug will be studied in your pharmacodynamic during your pharmacodynamic studies. So moving on to pharmacokinetic, guys, as I said, particularly pharmacokinetic is nothing but your drug movement from right from your absorption till your drug gets completely eliminated from the body. Uh, mm, it covers absorption, distribution, metabolism, and extraction. Whereas your pharmacodynamic, definitely, as I said, it shows the biological response or biological process in which the body responds after impacting by a drug. And here's some differences. Definitions are there, like the rationale of for in vivo PKPD efficacy relationship studies in preclinical models are two. Base first is to provide the dose response relationship to the effects, the efficacy resulting from one to multiple drug doses, right from the 5 mg, as is given you previous slides example, right from 5 mg to so on, the different with multiple doses, and to establish, as I said, guys, the efficacy of your particular drug, which is should be in acceptable range. Right here in this graph, you can see with time your drug concentration goes on, and here you can see the three colors here. One is yellow, green, and your the upper is your red. So the yellow range, what you can see is generally called as a subtherapeutic range, and the green one is called your therapeutic window or your therapeutic range, and the above that is your side effect toxic level or toxic window, what we call it as. So the drug concentration which remains in this green area will be known to give a good effect to the body. I mean, no toxic levels there and no subtherapeutic level. So if your drug concentration is still in your yellow range in your bloodstream, then there is no use of your drug, right, to show the action at all. So we required a drug which is in therapeutic window to show the action or to treat a disease or to prevent a disease there. So we always aim when we are doing when we are proposing a concept, or we are starting from I mean the concept of studies, then we will always aim that our drug should remain in therapeutic range to show the action. Here, there are some more examples here, like when your drug taken at zero, and it slowly tends to absorb in the body. With different parameters are there, we will be discussing in the coming slides. So once it reaches the maximum concentration, then it's the highest concentration. That is the point where you call it as a C max, the highest concentration of the drug that is there in your blood. Right. Slowly now, the once your drug gets Cmax and maximum concentration, the natural process is like your drug now slowly tends to get inactive, and it will come under the process for elimination. So the point from Cmax to T half, where your half of the drug reaches to half of your concentration, then definitely you will be called it as a T half. That is your half life. And further, you can see the point that the third point, C minimum. So C minimum is your lowest concentration of your drug in the in your blood or in your body. So where it has no effect at all. So it's an elimination process now. So the whole idea here is like AUC, area under the curve. This is what we want in all the particular drugs. Right, guys. So moving on, like step by step, we will be moving. Like absorption, as I said, what is absorption? Is nothing but the movement of drug molecules or drug particles across your biological barriers from site of administration to the blood stream. For example, if you have taken a drug orally, right, it starts absorbing right from your buccal cavity. Right. It reaches the gut, the stomach lining, and from there, it reaches the absorption from start from there itself, and it has to cross different barriers, skin barriers to reach the blood systemic circulation. So the process of movement of drug molecules across biological barriers from the site of administration till it reaches blood stream is generally termed as an absorption. Whereas, during this process, during this absorption step, some of the amount of drug will reach, instead of going to systemic circulation, some amount of drug get into the liver, where it get active form into inactive form. So that is called first pass metabolism, and that directly eliminates get out of your body, and that is called your first pass metabolism. Right. And there are some important points here, like for the absorption of a drug, we required a proper environment, like your gastrointestinal tract of your pH of your gastrointestinal is an important role factor for absorbing your drugs there. So pH of your GI plays a major role during your bioavailability. For acid, for example, here for acidic drugs, the drugs which are in a form of acidic form, so better absorbed at a low pH. So with high pH, basic drugs as vice versa. For basic drugs, you require high pH. For acidic drugs, you require a low pH for its better absorption, for better availability of your particular drug. And for proteins, agents that are minimally protein bound penetrate tissues, better than those are highly bound substances. Moving on to distribution, guys, here, once the drug is absorbed, right, it reaches the it has to reach the blood circulation. So before blood circulation, it has to distribute from different from different tissues till it reaches the blood. Till it reaches these tissues to bloodstream. So we have a barriers here which hinder the distribution rate. And the definition comes in this way, like the process of delivering a drug from bloodstream to the tissue of the body. So where it has to show the actual action, where it has to reduce the pain, where it has to reduce the burden of your disease, where it has to cure the thing. So that particular site is generally termed as your tissue or the site of action. So it has to reach from blood circulation to the tissue, is generally termed as your distribution. Moving on, guys. So as absorption rate factors affect, I mean, there are different factors which affect your absorption. Similarly, your distribution can also have a high distribution level or low distribution level, or there are several factors which can affect or impact your distribution level. So these some of the examples are like your tissue permeability of your drugs. For example, physicochemical properties like your molecular size, partition coefficient, and physiological barriers like your blood-brain barrier, your CSF barrier, your placenta. Different barriers have their different affinity to absorb and distribute the drug to the tissues level. The other are like your organ tissue size and perfusion rate and binding. Yes, this is again important. Binding of your drug to the tissue component, binding of your drug plays a major role in distributing. and and some other factors are like miscellaneous factors, or like your age, if you're pregnant, if you're obese, if your diet is completely different, I mean, abnormal diet, you take a lot of junk foods, then definitely your distribution rate gets matter there. Your disease stage, your drug interactions. Here, I will tell you the examples why, what are the main factors why our drugs get affected while distributing. So, as I said, like guys, during disease stage, like you can see when a patient has a liver issue or a kidney issue or whatsoever. So, he, as- for example, if a patient is suffering with liver disease. So in that case, your drug will not be, as you know, guys, your drug will be metabolized from inactive form to active form to inactive form by using lot of enzymes which is excreted by your liver. So liver plays a major role for converting your active drug to inactive drug to active form. So if the patient itself have a problem with the liver issues, you are definitely you will be your distribution of your drugs will get hampered there. Likewise, in big scenario, I mean, in clinical trials, particularly in phase 1, 2, you will be giving a single drug, and definitely you will not have other drugs. So it's simple. This is a straight away thing. But when you actually come into the market where you have a lot of patients, taking a lot of drugs for different indications, the same patient may have diabetes, hypertension, kidney problem, a lot of other diseases for which he will be using minimum, I mean, 10 to 15 pills per day, and per morning session 3 to 4 pills, per afternoon 2 to 3 pills, again for evening 2 to 3 pills. So taking altogether these pills, so may have drug interaction, drug food interaction. Again, it may escalate your distribution, or may de-escalate your, I mean, your distribution rate may go low with this parameter. So drug interaction, drug food interactions can have a factor. These are the factors which will distribute your affect your distribution of your drug there. So moving on to metabolism. So once a drug has been absorbed and distributed in the body, now it has to get metabolized from active form to inactive form, which is acceptable by your kidney, so that it can push your drug out of your body. So it want in this way. I mean, kidney want molecules in where the kidney can recognize that molecule, and it can excrete from the body. In similar way, so, as I said, liver plays a major role here for converting a to drug to active or inactive form to active form. And here, most of the drugs use cytochrome P450 enzymes for metabolism of the drug here. So again, here factors which can hamper your metabolism is like, again, if you have a disease with your kidney disease, if you are suffering with liver, if you are suffering with other kind of diseases, then definitely you will have impaired, or the drug can show improper metabolization. So here, the overall, you can see how the metabolism actually works here. When you take a drug, it reaches the gut, and then bloodstream, where it will bind to receptors, and once it's bind to the receptor, that receptor will take the drug molecule to the site of action, and at that point you can see the effectiveness of your drug, or side effects once it reaches the system. I mean, the site where we want the drug to be act. So there you can see the response of your drug, and definitely once it shows the action, definitely it has again have to go back to the bloodstream, from where the liver get your active form to inactive form, it get metabolized there, and then finally step is your elimination. That is pushing the drug out of your body. So excretion. So excretion is nothing but, as I said, the process that ferment removes your drug from the body. So all drugs are eventually right eliminated once. If you have taken a drug, some drugs may have better half life, like 1 to 2 days, 3 days, up to 72 hours. But there are some drugs which remains in the body for a couple of weeks as well. So different drugs have their different half life based on their formulations effect, and based on the compound of your particular drug. So it has different time levels there. But anyway, at the end of the day, the drug has to get eliminated from the body completely. That process is generally termed as your excretion wise. So here most of the drugs, particularly water soluble drugs, and their metabolites are eliminated largely by your kidneys through urine, and therefore a drug response depends largely on kidney function. Once you go to the physician, right, guys, this is like in the real practical way, I'm trying to explain you. So when you go to the physician, definitely when if you start prescribing a drug for any particular disease or indication, before prescribing, he will definitely ask you whether you have a kidney problem, or do you have any liver issues, whether you have any cardiac issues, before he prescribes the drug, so that the drug, if it remains more than the estimated amount worth it, then it will have the toxic effect. So, right. So, particularly physicians are more concerned about the elimination of the drug, because the toxic levels can have a very unwanted effects on your body. So, that's why before prescribing a drug, your renal clearance, your EGFR clearance, a lot of parameters are there. Your physician will calculate based on that. He will decide the dose, what actually you need, and based on your health condition. So moving on, your elimination process, guys. Like as I said, through kidneys, majority of your drugs get eliminated from urine via kidneys. But we have other forms of excretion. I mean, systems where you can excrete your drug from the body, apart from your kidney and urine. For example, from your saliva, your sweat, like breast milk, your inhaled exhaled gases can also are the routes where you can excrete your drug from the body. So that is the reason, guys, when your I mean, when woman is lactating, definitely your doctor will ask whether your mother is feeding. If feeding, definitely some of the drugs can cross the and reaches to the milk, and it can it can carry the effect to your baby as well. So some of the drugs are contraindicated when the mother is lactating, then pregnant woman is there. So in that scenario, definitely there are a lot of choice of drugs are there. So which does not cross blood-brain barrier, which does not. I mean, there are a lot of several drugs. Here. I mean, based on the scenario, real practical scenario, we will choose the drug for giving the patient. And I mean, the factors, again, as I said, if a patient is obese, if patient is like having renal failure kind of issues, or if you have any liver problem, if you have any, so that the similar parameters can also affect your elimination process, guys. So these are broadly how you understand the pharmacokinetic and dynamic parameters. So now, as we have seen in the previous slides, to study the toxic levels, we require animals, right? We required some cells, we required some animal models, we required some computer simulations to know the effect of um, the drug, how it works, at what concentration, at what is the highest dose where it can show the toxic level, where it can show therapeutic range, where it can show sub-therapeutic. As you can see, guys, like, did you see, guys, there are a lot of drugs which just comes and sedatives. The concentration of drug is in points 0.25, 0.5, 1 mg. So that amount, 0.25 amount of drug will have a lot of effect on the body. Whereas you take the metformin, 500 mg, 1000 mg, right? So at that level, you require to neutralize your blood sugar levels. But whereas you are to get the sedative effect, you just require 0.25 mg. So you can see this is a huge difference. So I mean, again, this is based on the compound. So what you are in a developing stage, or the compound which shows the action. So in this, I mean, particularly in this market, you will be having a lot of different strength of your drug, 5 mg, 10 mg, 15 mg, based on the body mass, your height, personality, your disease condition. You'll be prescribed with a different dose, to reduce the toxic levels, and to have a good. I mean, you should be there in a good therapeutic range. So as I said, guys, to know the toxic levels, we will be using animal models or animals there to know the effect. Whereas to study the parameters like pharmacokinetic parameters like absorption, distribution, metabolism, and excretion, you required a bioavailability study, right? What is bioavailability study? Why we will do it? And we will be discussing in the coming slides. And what is bioequivalence studies? Why we will be doing? We will be seeing in the coming slides. So moving on to BA/BE studies now. So bioavailability, as says the definition, the fraction of your drug administered that reaches the systemic circulation. So whatever drug which reaches the systemic circulation, that particular amount of drug is nothing but your bioavailability, which is there in the bloodstream. For example, if you have taken 100 mg of a particular X drug administered orally, maybe 70% may reach to your blood circulation. So now the amount of drug you have taken is 100 mg, but the amount of drug which reached the systemic circulation is 70%. So the availability is now, your bioavailability is now 70%. The 30% may have the first pass metabolism effect, where directly your liver will convert your drug from I mean inactive form, and it will excrete the drug from the body, and the remaining 70% reaches the systemic circulation. That particular 70% will show the effect at the site where we reside site. So generally, bioavailability testing generally required for new drugs, which are completely new drugs. I mean, completely for new drugs. For example, for your bioequivalence, as I said, by BA/BE. So BA will be the bioavailability will be checked for majorly for new molecules, for new drugs. Whereas your bioequivalence, here, nothing but when you are formulating the same amount of drug into different formulation, right? Different formulation. Like, for example, from from tablet form to syrup form, from syrup form to some other form. So when you are creating a new formulation to the drug, and you are copying the particular drug and you're producing a new drug, in that way, in that scenario, we will show the equivalence, whether the master drug or your standard drug is same as that of your the present drug. The comparative studies is nothing but your bioequivalence studies. By definition, here is like your comparison of your bioavailability of two drugs or more product formulation containing the same active ingredient. Here, main point is we are using the same active ingredient, but you may change the color, you may change the size, you may change the other excipients like coloring agent, um, emulsifying agent, the dissolution. There are a lot of other parameters. Other ingredients when you are formulating a drug. So if you are using that, then definitely again you have to test whether in that excipients your bioavailability is same as that of your standard drug or not. Moving on, guys. As I said, like when you are doing BA/B studies, you will be having a reference product. Yes, generally, you call it as a regulatory authorities designated reference product, and it's a global innovator product, and it is protected by the patent and marketed by the brand name. And efficacy and safety profile is well documented and extensively trials has been done. And Whereas in your generic drug, you have to show the bioequivalent, and you have to follow some rules and regulations here again, which is described by your CDSU. And so the innovator drug is nothing but your new drug, which is completely new drug, is called it as your reference product or your standard drug. If you are copying the same drug after patent is completed, like you have a lot of generic drugs now in the market. Our Prime Minister, Mr. Modi, also supporting. I mean, is creating awareness among patients that push yourself to generic drugs. Is what are generic drugs? It's nothing but once a drug is completed with their patent years, like for 15 years, 20 years, then the other pharmaceutical companies are now legally produce the same active ingredient with different inactive ingredients and at a lowest cost so that it reaches the patient. Like the copying is nothing but your generic drug, and the copying is the drug from where the standard product is your standard drug. So generic drug, as I said, guys, which is identical or bioequivalent to your brand or reference drug in terms of active ingredient, route of administration, dosage form, strength, indication, and safety. So you will not compromise when you're copying a drug in these areas. Like active ingredient will be same, strength is same, dosage form is same, safety parameters are same, and route of administration are same. So may have different parameters like inactive ingredients, like as I said, their color, shape, their preservatives, their binding efficacy, all these other parameters which are used apart from your active ingredient are considered inactive ingredients, and it may differ. You can use based on your own formulation. If you want a good formulation which shows a quick action, like a drug which is a simple paracetamol compared to your paracetamol SR, sustained release. So yeah, added some binding agents which sustainedly shows the action throughout the time. Whereas your a simple paracetamol, it will directly show the effect and it goes on. But in whereas a sustained release or long-term release kind of drug formulation, once you take one single tablet will suffice throughout the day. Like if it is a sustained release, so it will release a drug slowly at different time interval. Whereas your other drug, definitely it will show the complete action in one go. and it will get rid of your body, and the action is gone now. So if you are using different formulations, that is okay. But your active ingredient should not change in generic drug, and strength should not change, dose should not change, and definitely safety we will not compromise at all. Almost you can see now, and half of the market is full of your generics now. So most of your actual innovative drugs, standard drugs, patents years are completed. Now every pharmacy, the newer drugs are coming. Hardly you can see the newer drugs in the market now. We are just copying. For example, like antibiotics, you see a lot of lot of conferences, lot of efforts from the government is they are putting to reduce the antibiotic resistance, because we are told now we are resisting to antibiotics. Even high-end antibiotics like meropenem and all that are getting resistance. So that is the level. We need new drugs, new antibiotics with new drugs to get rid of this resistance kind of thing. So particularly in that scenario, definitely. So as out of the topic box, I would suggest, like, guys, whenever from your parents or your relatives, you should tell them that antibiotics don't use without a prescription and should be used in the time duration what physician or your doctor says, and it has to be monitored well to avoid further resistance, because we are dealing with a lot of antibiotic resistance cases now. So moving on, guys, as I said, for everything we need particular guidelines, particular approvals from different regulatory authorities to do this kind of studies here. So one such if you are carrying out these studies, bioavailability or bioequivalence studies, if you want to do it in India, definitely you have to take permission from CDSCO. Previously, we used to follow the Schedule Y guidelines, but right from 2019, the guidelines has been replaced with NDCT 2019 guidelines. So now we are not using Schedule Y guidelines. Instead, we are using NDCT 2019 guidelines for conducting clinical trials and preclinical trials as well. So you can download, you can just Google and type NDCT 2019 guidelines in your URL, and you will find a PDF, which is a huge document given in two languages, English and Hindi. And the first part is Hindi, the second part is again in English form. And I would suggest you download that PDF and you just keep on reading that particular guidelines book. Definitely it will help for your this thing for your clinical trials. So here's some, again, to understand in the better way what is generic versus your brand drug or innovator drug or your reference drug. So here you can see when you are doing a studies, when you are doing a trial, so if you are innovating a completely new drug, what are the requirement studies? What are the requirements to get a marketed drug? So here you will be doing animal studies, clinical studies, bioavailability studies. You will be establishing their chemistry, their manufacturing process, their labeling, testing, and all that stuff. So you will be required all this step to establish a new drug, your brand drug, a completely new drug. Whereas if you are just copying a drug after completing of your patent, for example, 10, 15 years after that, if you want to just copy paste your drug with your name, with your brand name, you can do that with your generic drug. And here, what you require is the application will be your abbreviated new drug application. Whereas if you are submitting for a new drug, then definitely you require new drug application. And ANDA is for your generic drug. Here you can see we are not doing animal studies because we have already established the efficacy safety parameters. So we don't require any more testing now. So clinical studies are also not required because we are not testing we are not changing the active ingredient here. So definitely not, we will not be doing clinical studies like phases 1, 2, 3, like bioavailability studies, because it's already established the bioavailability, absorption, distribution, metabolism, excretion parameters. So that data will be copied and will be submitted as such for ANDA application. The only change here we can do is that some chemistry form, the chain can be changed so that it shows the quicker action. So manufacturing can be changed by using different inactive ingredients, like different labeling. You can see the brand comes in different color, different shapes, strips, and all that. That can be changed and it can be submitted as a generic drug. So moving on, guys, to do this, to do this bioequivalent studies, we required, a good setup to establish, to show that your generic drug is same in terms of safety and efficacy in with reference to your product. Reference drug. So we have a lot of again a process, stepwise procedure, as we do in clinical drug development phases like phase 1, 2, 3, 4. For bioavailability studies, bioequivalence studies, we again have different steps involved for doing bioequivalence studies. Before moving bioequivalence studies, there is an important definition should be remembered by you guys. What is washout period? There's nothing but, you give two drugs. With in between two drugs, you should be giving a good time interval so that your first drug completely get eliminated from your body. So that when you're studying at second interval, when you're studying in the second term, the previous drug carry on effect should not be there in your the coming thing. So a proper time should be given. So the time between your two treatments is called your washout period. So definitely, as guys, uh, that as I said before, like food plays a major role. Before food and after food, food can also interact with your drug. So we have to do some of the drugs which required to be given in fasting. We required fasting studies. Some of the drugs which are given post meal, that is post fed, then the criteria of your drug, the concept is your post fed, after food. So if you are doing fasting studies, drug trials like overnight 10 hours fasting is mandatory, and for multiple dose study, 2 hours fasting before and after. And for fed studies, drug recommended with the food and modified release product. Different food have, different states, different countries have their different food intake options are there, right? Here, India itself, South are more prone to have idli dosha kind of things, whereas compared to North, where you will have poha. So that food can also have the effect in absorption in bioavailability. So for that reason, we need to do the bioequivalence studies to establish the availability of the drug. So to conduct such studies, what all things we required is your pre-study requirements. Like as you do in clinical studies, you require ethical committee approval. For animal studies, you require animal ethical committee approval. Here also you require same institutional ethics committee approval to conduct your bioequivalence studies. And definitely you will have written procedures how to proceed. You'll be doing your studies stepwise in accordance to your ICH GCP guidelines, CDSCO guidelines, your NDCT guidelines. And when you're doing when you're doing the study, the area, infrastructure should be adequate infrastructure you require. A good clinical facility and your trained personnel are required. Qualified persons are required to conduct such studies. And for bioequivalent studies here, one important interesting point here is like for bioequivalent studies, bioavailability studies, you require only healthy volunteers. Why this important point here? Like, see, if you're testing the drug uh, in a person where he has a lot of diseases, like we already discussed, like renal failure or kidney failure, liver failure kind of things, that will hamper your absorption rate, and ultimately your outcome will not be good because you're having some kind of disease. Whereas in healthy volunteers, your heart, your hair, your liver, your kidney are under good condition. They are working well. They are within the normal ranges. In that scenario, you will be actually knowing the correct the points and the correct parameters, the correct results. Like for you're working your particular person liver is working well, so definitely metabolism excretion. I mean metabolism of inactive to active form to inactive forms are well and good there. So there is no problem at all to get the data from that particular patient. Whereas disease patient, you're not able to get the actual data, correct data there. So we required healthy volunteers when you are performing bioequivalent studies. Except some drugs are there like cancerous drug, high end toxic drugs, where we cannot expose these drugs to healthy volunteers and make them that ethics will not allow you to do that. So in that scenario we may use a direct patient itself to do DE studies on that particular indication, like for example cancerous studies. Moving on, guys, to do bioavailability or bioequivalent studies, we have to plan well before you initiate the study. And four major important parameters are required here. Like your doctor, I mean, the particular physician should be there to conduct the study. Your protocol, which shows the complete stepwise process, and the objectives, what all you want to achieve. That objective should be well established and described, and design, yes, definitely. You will have a designed which is already established to get the data. For example, your I mean for fasting state or post meal or analytical methods, you should be well designed before you initiating studies. And finally, without the population, without the subjects, without the patients, you will not be able to complete the study there. So study population is must. So here, overall view of BAB studies, like we start with the study design, and then we develop our documents. We will submit to regulatory authorities. One, we get the approval letter. So the site selection happens. Then ethical committee approval goes on parallelly, and then there will be a there will be a time where we will test the drug to human beings, and then we monitor. And definitely, we will be taking the parameters at different point levels. We will be collecting the data from the patient, their side effects, their adverse effects, their serious adverse effects, their therapeutic effect, and all that will be recorded with timingly. We will be following up with the patient like for 24 hours, whatever. I mean, based on your drug half life, your follow ups are designed and during in your protocol time itself, and definitely that data will be gathered and statistically be analyzed, and we will make a final report, and that report will be submitted again to regulatory authority for to get the approval to the into the market. So, what are the three major factors that affect bioavailability? Now, guys, pharmaceutical factors like physicochemical properties like formations and all that stuff, pharmacological parameters like patient-related parameters, as already discussed, like disease condition, route of administration. Yes, definitely, your parental route are much faster than your oral route. So, likewise, different routes plays a different bioavailability. Like here in this particular slide, you can see that uh, you know different parameters, different formulations have the different bioavailability rate, like oral, parental, transdermal, your sublingual, your rectal, your inhalers. A lot of formulations are there. All these formulations have their pros and cons, their difference in bioavailability and efficacy point of view. So coming to pharmacodynamic, so dynamic is nothing but how your drug shows the action. What is the mechanism of action? What are the receptors involved in the mechanism where it shows the action? For example, how the drug binds. to which particular site, to which particular receptor, whether it is extracellular or intracellular, so that you will be seeing the response. The response may be stimulation, inhibition, it can be depression, it can be replacement, irritation, toxicity, whatever. I mean, if a drug given, it shows the action, right? So what type of action, that definitely you will be knowing. For example, you have a headache, you have taken a Dolo 650 mg and you're all right after 1 hour, 2 hours. That means your drug is actually working, right? So, but the action behind that. So, what are the how the drug got absorbed? What are the receptors where it got binded? How it reaches the site of action? That particular area is nothing but your pharmacodynamic. I don't want to go much deeper into that because has again subtopics in that. But overall, is just a mechanism of action. I will just end up my today's topic with one classic example here. Like a receptor. Here you can see lock and key kind of mechanism of your insulin. So here the patient has high glucose level in the body, and once the insulin is given, that will, I mean, that will reaches the systemic circulation and at the site of reaction, and we can see in the cell there are a lot of glucose molecules in that, and once the insulin bind at correct receptor, it will reach the glucose molecules, it will bind to that glucose molecules, and it will excrete that glucose molecules through urine or whatever. And that is how the insulin works, and that is how your sugar levels will come down. So this is again a big topic, guys, but overall view, you have to understand what is difference between pharmacokinetic parameters and dynamic parameters, and to assess this, what all you required bioequivalence studies and bioavailability studies, what are the generic drugs and all that stuff, we have already discussed. So with this, I would like to thank you guys for your patience listening, and hope you understood well. And if you have any questions, please raise your questions at any point of time. We are there to help you guys. And with this, I would like to end this session.